Good. Hello, good afternoon. Now I'm very excited to, in the next 20 minutes or so, talk to some extent on how we actually leverage big data to understand how pathogens spread and how that also can have an impact on, well, all of us, society, what knowledge we can actually gain and translate into actionable future doings. So first, all life is characterized by the genetic code. Every one of us has in every single cell 3 billion so-called nucleotides, those ACGTs, and the particular order of those nucleotides in your cell determines, say, if you have brown hair, black hair, blonde hair, and other characteristics. And within one person, all the cells have exactly the same sequence of nucleotides, up to maybe a few, say, errors. But between us, the order of nucleotides is slightly different. That's why, for example, we may have different hair colors. And as much as all of us and all living organisms are characterized by this genetic code, also any pathogen, including the viruses, where it's being controversially discussed, is that live or not? They also are characterized by a genetic code. And why it's discussed if they're live or not, which we're not doing here now, is that they can't replicate on their own. They always need us, say SARS-CoV-2 needed us to replicate and actually spread. So, for example, being with SARS-CoV-2, instead of 3 billion of those nucleotides, that virus is just characterized by 30,000 of those nucleotides, so 30,000 AZGTs. And also it's one variant just has one such sequence, not like us, many cells. And this pathogen genetic code is actually very relevant to all of us. So first, to us scientists who want to understand pathogen evolution, of course, only if we know this pathogen code and also how it's changing through time. So is the virus a year ago, was it looking different from today? That tells us about the evolution. And in the pandemic, actually, that was also then knowledge which was often highlighted in media. For example, based on looking how this genetic code changed in SARS-CoV-2, scientists came to the conclusion that certain variants, say, were more transmissible, causing a faster spread of the pathogen. That directly goes then into public health in a way. How do we respond to that pathogen spread? So maybe new measures are needed, but also, or in particular, industry. So based on knowing the pathogen genetic code, the pharmaceutical companies adopt their vaccines such that the new vaccines match well to what is circulating currently. And over decades, that has already been done for influenza. So every fall, if you get the vaccine shot for influenza, it's slightly different from the vaccine shot you would have gotten the year before. Why is that? Because influenza continuously changes, and we want to, vaccinate everyone with the vaccination which is as effective as possible, meaning being matched as good as possible to the currently circulating strains. And finally, that's always being done in the pandemic. Then this pathogen code even made it in a lot of, or was politicized, I would say. So Johnson then saying that there's a deadlier variant, but then scientists contradicting that statement, that it didn't see the databases. So for a while it was, I guess, everywhere. But as a scientist, public health and industry, we worry about this genetic code all the time, with the aim that to make a positive impact for everyone. So traditionally, actually, every year, people around the world sequenced influenza. And the main motivation was actually to update the vaccines alongside understanding better influenza evolution and potentially, adapting treatment schemes, etc. And so overall, before the pandemic started, there were roughly 300,000 influenza genomes shared, which were globally available. HIV is the other pathogen which was traditionally sequenced a lot. the reason there is that for diagnostic purposes, if somebody is infected with HIV, the medical doctor wants to know what is actually the genome of this HIV, because if there's certain mutations, the patient might not react to particular treatments, meaning have a resistance, the virus has a resistance to a treatment. And to determine the optimal treatment regimes, it's beneficial to actually sequence the virus. So that has been done, say, in particular the global north quite a bit. And so roughly one estimates there's 1 million such HIV sequences available. That's an estimate because with HIV, for ethical reasons, one cannot openly share all the data at all times because it's a chronic infection. In theory, you can go back from the pathogen to the human if you have the right identifier. And then 4.5 years ago, the pandemic started, and oops, that's what happened. Within, essentially, that was in the first half of the pandemic, within 2.5 years, 17 million of sequences were generated. So that was compared to anything which was available, a huge jump, and actually then, of course, also created challenges, how to actually handle all this data, what to do with all this data. In many different countries, researchers, public health, they sequenced the virus to understand, are there new variants, would there be a new variant of concern, and then shared this data. So we set out, generally in our research, we want to understand globally what's the diversity of a particular pathogen. For influenza, it was relatively simple, 300,000 genomes, I mean, that we can roughly handle. Now for 17 million SARS-CoV-2 sequences, if just naively downloading and naively storing, that would be 500 gigabytes of data were naively stored. I mean, you can say, you can of course store that, no problem. You can also compress it further, then it would be around 1 gigabyte. So no problem at all, you can put it on your USB stick. However, this data, if it shall be useful for public health, we actually need to be able to query it all the times and ideally in real time. And so if you want to query the compressed data, while the 1 gigabyte you could also have in RAM or in fast storage, actually you have to decompress each sequence, ask is there a particular mutation, and compress it again. So this break was probably annoying. I want to illustrate. That was a few seconds. That's how long it takes to actually ask, you know, does a particular sequence have, or how many sequences in Switzerland have mutation X but not mutation Y? If you do that once, maybe you can live with those few seconds of annoying silence. But if you have a dashboard and you want to query and explore the data, you soon just stop using this dashboard because you want to have answers right away. And so typically the dashboards becoming available for those 17 million sequences showed you only pre-calculated statistics. So things the people providing dashboards would think are relevant to you. Say, you know, when was the first Omicron sequence detected in Switzerland, and how many did we have by the end of 21, or things like that. But actually, we have much richer information in the data, and if we want to leverage it both for research and for public health, we want to access all the information. Oops. So what we were then doing, we actually developed a sequence-specific compression scheme. And the sequences, I mean, it's very much, you have the rows, the ACGTs, and the columns are the different sites. And so we came up with a compression scheme where essentially all the information was still encoded in a data. So we still had it compressed to 1 gigabyte, so you can have it on very fast memory. And because you don't have to decompress, you can actually query in milliseconds all the data, all those 17 million sequences. And so this compression scheme, then we actually used in with a front end we implemented. It's called COF Spectrum, where now you as a user can query for any questions you have about this 17 million sequences. How many mutations in which geographic locations, at what times, and what was the frequency, etc. And you get plots as I show here. For example, right now, this plot shows the current circulating variant, KP3 it's called, don't have to remember, it's just one of the many variants for SARS-CoV-2, and it just increased in frequency. And we have that all the time, probably in a month. It's not KP3, but KP10, and you again see it increases in frequencies. However, of course, for public health industry and research, it's important to see what is exactly around right now, and when there are decisions on vaccines, it's important to acknowledge which mutations the vaccines should potentially target. And so we launched Prof Spectrum initially. There was still a bit less data than 17 million, pretty much early 2021. And then it has been used by researchers and public health officials to query the sequence data. And in particular, it became valuable to propose variant names, lineage designation, we would call it. Because if we want to talk about those 17 million sequences, we actually want to have names for clusters of those sequences. And there's international efforts doing that, defining that. We are not involved in defining precisely which cluster we now give a particular name, like Omicron. But with our tool, you can track which clusters increase in frequencies, etc. And so proposing what could or should potentially get a name. And then vaccine producers in meetings use then the tool to propose which variants they actually think they potentially would want to look at and adapt the vaccines in that way. What we actually used the tool for was the first time we used it for some, public health question was when we just launched, so the beginning of 21. And as you may remember, in the beginning of 21, we just came out in Switzerland of a terrible fall wave with many people dying of SARS-CoV-2. And let me see, yeah. And so this was the wave, November, then we had falling case numbers. And then just around Christmas, we got those worrying reports from the UK that there's a new variant and it's faster spreading than the previous wild-type variants. And indeed, in Switzerland, we then also saw digging into the data and actually asking Prof Spectrum, how fast do those particular mutations, this combination of mutations grow? From pretty much nothing within just 2 weeks, essentially, it was already at 3%. So it seemed it's really accelerating the pandemic. And indeed, we then also fitted mathematical models. They indicated that this alpha, which it was later called, has a pretty big transmission advantage, 40–50%. So if, I would be infected by a wild-type virus, I infect 2, all of a sudden I would infect 3. So then instead of going from 1, 2, 4, 8, etc., it goes from 1, 3, 9, 27, so the growth would be just much, much bigger. So based on those calculations, we actually informed through the science task force the government, or they actually ask pretty much daily around Christmas, New Year, what's going on, is there another wave? And so we said, well, yes, it all looks like in March we'll have another wave because then alpha is dominant. And so they actually, if you may remember, in January we had a period where only 5 people were allowed to see each other and masks in all shops, etc. This was in a period when case numbers went down. So many people didn't quite understand, well, I wouldn't understand either just looking at this plot, why would some government strengthen measures? But it was because underlying there was an increase in a variant. And actually later, that's how the increase continued. So yes, it took over in March. It was dominant. There was still a bit of a March wave, but one estimates it was dampened by quite a lot because all of us had less contacts. We used that in particular for Switzerland. Other, as said, researchers used it for variant classification. However, we see such tools to employ those 17 million of sequences much broader. Ideally, it contains sequences from any country in the world to sequence, so more than 100 countries, and public health officials in those different countries should be able to query that data. First problem, our dashboard is in English. Not in every single country, every health authority, you would find very good English speakers. And second, you need to query in a certain technical terms, a bit like SQL, which also, if you are in a pandemic, overwhelmed with everything, maybe you don't want to learn exactly how we query our data. So we thought what would be great would be a chatbot around the dashboard and the data. So sure enough, we actually used GPT-4, or first GPT 3.5, but then when GPT-4 was available, used that to actually translate a question the user types in. So which are the 5 most often sequenced variants in Europe outside of Switzerland, say, this year? Then the chatbot translates that question into a query for the database. Our database is being queried, gives back, the answers in a table, and then actually also the answers with the GPT-4 protocol are translated back into a text so the user can check is the answer actually corresponding to what I asked, to minimize the risk that the chatbot misinterpreted the question and gives back something completely different. So with natural language, you can check back what's happening. And then we actually had users from very different language backgrounds using it. It was it's still a prototype study. So a couple of messages they put in, and we checked, how often did they actually get the right answer. Remember, our dashboard is in English. Behind the database is just a database. But because it's GPT-4 empowered, it can, of course, deal with all the different languages. In particular, GPT-4, Swiss German, which is not even a written language, but of course in the internet you find Swiss German, 18 out of 18 questions were correct. So we see that as an opportunity to actually bring kind of the information of those huge data sets globally to public health institutions. However, I should say, right now, it's still a prototype. I wouldn't advertise it as, everybody should now just have the chatbot and do it that way, because very often questions can't be translated back to the query. You pretty much need to know the background, how is our dashboard and our querying working, that you can actually type the question in a way that it's reliably translated back. So it needs more investigations. I think it's powerful, though, to have those large language models coupled with exploring the big data sets, which I guess otherwise you need data scientists to really make sense of this big data. And actually in Switzerland, also there's this project Statbot.Swiss, which aims to develop a chatbot to interact with Swiss Public Health Administration's open data. They just released a preprint, and I guess they also had a bit of challenges like us. We were very motivated and still see it as the vision to get there, but they are saying in their preprint, our experimental analysis illustrates that current large language models struggle to generalize well in generating SQL queries, so they do it also similar to us, on our novel bilingual data sets. So I guess it's fair to say different entities have the different vision to use chatbots for those big open data sets, but it's not in a production mode yet, but more in a prototype mode. The second direction we went from our, way to compress the data and query the big data was that we thought we should not just do it for SARS-CoV-2. In the future, I'm convinced now that also sequencing is easier, cheaper, we'll have more and more sequencing data for many, many different viruses. So we actually put then the advances we made for SARS-CoV-2 into a more general software package that anybody, say if you host HIV data, which you can't even share for ethical reasons, you can actually host them in a database where you can have a facilitated way to query and manage your data. We called it Loculus, and as said, it can facilitate local but also global data sharing. And there I'm at the last part of my talk. All those big analyses are only possible because actually people around the world shared 17 million of sequences. However, the crisis also showed that none of the data sharing platforms people are really enthusiastic about. Because either they have certain technical challenges. It's super cumbersome to upload your data. You pretty much need somebody to worry about that quite a lot. Or then there are really tools to adapt it to pathogens, but they have a lot of governance issues, transparency issues run by a single person, not from some public funds. So people more and more were unhappy and also then didn't share for, even though it was for public health, because they were worried about being scooped in their research. And so now we use our software locally, so which all came from, how to manage large number of sequence data, into a public data sharing platform, pathoplexus.org. I invite you to visit this. Currently for viruses which cause very deadly infections. Ebola, West Nile, you've probably heard about, Cameroon, Congo, hemorrhagic fever, so that people can actually easily share data there as a prototype to, in the future, hopefully advance, get much broader, and cover the pathogens we are all of interest. We right now don't do it, say, for influenza, because we first want to have this more as a prototype, see how it goes, and then once we're really sure everything works super well, we go to the more pathogens where more sequences are being submitted. And with that, I hope I have shown you that with this explosion of data, we have a lot of opportunities. We now have the data for all those computations people were talking about earlier. But we also have challenges, like, posing new research questions. As scientists, I would say it's even more opportunity. But anyways, how do we manage this data? How do we query it? We required different computing infrastructure when doing those studies. So I guess with all the big new data, we also have other opportunities, challenges. And with that, I want to end with acknowledgement, all our funders, our team, and thank you for your attention.